Transcription. Transcription converts a gene into a single-stranded RNA molecule. All right, so what we're saying here is that in the nucleus, we have DNA. All right, so DNA is stored in the nucleus, as we know. What we just talked about recently was that DNA is replicated. We kind of went through this long description about how DNA gets replicated. Okay, well now what does it do? All right, well, so the DNA, what we need it for is it is a blueprint, or it's a gigantic book of information that we need to read so that we can ultimately make protein. Because protein, like I told you, is the magical molecule in the human body that does an unbelievable amount of things, almost everything you can think of. So anyway, we've got our DNA, we need to get to protein. It's not just a straight shot, okay? In fact, we have this central dogma that states, so if you're following along at home using your power notes, this is what you want to pay attention to. It states that first you start with DNA, number one, and then the second thing that happens is a process called transcription. Transcription gets you to the third thing, which is RNA. A process called translation, which is the fourth thing, gets you to something known as protein. So really the central dogma is the DNA, RNA, protein, and these are the little processes, the name of the processes to get you to those things. All right, how is RNA different from DNA? Well, remember DNA. DNA has, so if you're making a list, you can look at your old power notes for what DNA has. Is it a ribose sugar? Is it a deoxyribose sugar? Does it have A, C, T, and G? Is it double-stranded or is it single-stranded? So you need to think about that. I will tell you about RNA though. RNA is right here. We have three things I'm going to tell you. So RNA has a ribose sugar, so that should seem different from DNA. RNA has uracil instead of thymine. So no thymine, no T in RNA. And then RNA is single-stranded. Okay, so now if we're looking at the steps of transcription, again, trying to help you with your power notes here, making it a little more, hopefully making it a little more palatable for you. Step one is, well, first of all, the whole process is catalyzed by this enzyme called RNA polymerase. All right, and that's going to be the big blob, super big blob there. All right, so there's all kinds of complex things happening. We just show it as a big blob. Other proteins are involved as well. Okay, so RNA polymerase, number one, and other proteins form a transcription complex. That's what this whole thing is. All right, so that includes the DNA molecule in here and everything else. The transcription complex recognizes the start of a gene and unwinds a segment of it. So in this case, this was the start site, and you can see how it's been unwound. Okay, step two in our power notes would be that RNA polymerase reads one side of the DNA template and adds a complementary strand, or excuse me, well, yeah, it is a strand, but adds complementary RNA nucleotides to make a strand. All right. Okay, after that, it doesn't. This doesn't really follow, step by step 3, 4, 5, 6, and 7 that are on your paper. So I'm just going to point a couple things out to you. Let me stay here, actually. This is a growing RNA strand. Remember that this, on both sides, is the DNA molecule. These are nucleotides. This part here, the whole thing, is newly synthesized RNA. Kind of a repeat of what I said earlier. We said it was a growing RNA strand, but this is the newest part of it here. And just the big old blob, remember, is RNA polymerase, which makes up the transcription complex. All right. Okay, let's shift gears here. And about ready to wrap this up. let's talk about, oh yeah, so in the end, the RNA strand detaches once the gene is transcribed, and we'll talk about what happens next in the next video. All right, but last thing here are the three types of RNA. Okay, you have messenger RNA, or mRNA, that carries the message that will be translated to form a protein. We have something called ribosomal RNA, or rRNA, that forms part of the ribosomes where the proteins are made. And then we have something called transfer RNA, or tRNA, and that's going to bring amino acids from the cytoplasm to a ribosome. So our next class period, longer class period, we're going to do a simulation of this whole thing using paper and cutting stuff out, all that good stuff. So this will totally, hopefully, make sense to you. And just to wrap this up here, transcription is kind of similar. They're saying it's similar to replication in that both involve enzymes and both involve complementary base pairing, A with T, C with G. But there's a different result. So in the case of replication, you wind up with all the DNA. All the DNA gets copied. And replication makes one copy, whereas transcription can make many copies. So let me rehash that one more time. Two processes have different end results. Replication copies all the DNA, whereas transcription copies just a gene, which is a segment of DNA. Replication makes one copy. Transcription can make many copies.